Welcome back, students. Today we are going to discuss part 3 of Unit 2, that is Unit of Life. But before I start the lecture, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of original creator and owner of these images. If you are the owner of any of the image used in this particular PPT or have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In today's video lecture, we are going to discuss about enzymes. What enzymes are, their chemical nature, their classification, mechanism of action, and all the topics related to enzymes. Okay. So, first of all, what are enzymes? They are biological catalysts. Enzymes are biological catalyst. And what are catalyst? Catalyst is defined as a substrate that increase the rate of a chemical reaction. Am I right? And without being itself changed in the process, right? Or without being involved in the process. They just enhance or increase the rate of chemical reaction. This is the work of any type of catalyst, right? So catalyst is defined as a substance that increase the rate of chemical reaction without being itself changed in the process. And enzymes are also a type of catalyst, or we can say more specific, these are biological catalyst or biocatalyst. Okay? But and all the enzymes are proteinaceous in nature, but there are few exceptions like ribozyme. Okay, ribonuclease P is another type of exception. So sometimes RNA molecules or we can say nucleic acid also act as a enzyme. Okay, and the enzymes are linear chain of amino acid which give rise to three-dimensional structure or globular form, globular proteinaceous form. Okay, so enzyme as a biological catalyst. Normally, in the absence of any enzyme, if enzyme is not present in any type of reaction, then what will happen? If we combine, if there is any reaction going on between carbon dioxide and H2O, and enzyme is not there, means uncatalyzed reaction is there, then only and only 200 molecules of H2CO3 being formed. And in what is the time? In how many times? It will only able to synthesize this H2CO3 in an hour without any enzyme. If a reaction is going on, as you can see in the x-axis, reaction process is there. This is your x-axis and this one is your y-axis, right? So if any reaction is going on between carbon dioxide and H2O and there is no enzyme in between, then means, or we can say the reaction is uncatalyzed. In that time, that reaction need so many times that reaction is very slow and only synthesize that type of reaction only 200 molecules of H2CO3. But if some enzyme like carbonic anhydrase is there, so if this particular reaction is going on in the presence of any catalyst, like in this case carbonic anhydrase, then this particular reaction speeds up about 6 lakhs molecules per second. Okay, so what does it mean? It means that this enzyme or biological catalyst actually accelerate the reaction rate about 10 million times. About 10 million times. Okay, as you can see over here also. How? What is the mechanism behind this? We will discuss it later on. Okay, but its presence means enzymes presence actually enhance the uh, um, activity type or product formation type, enhance or accelerate the reaction type or reaction rate. Okay, now catalytic efficiency. Now let's discuss some properties of enzymes. Its catalytic efficiency. These are highly efficient. Enzymes are highly efficient, 10 to the power 3 to 10 to the power 17 times faster than the corresponding uncatalyzed reaction. These are highly efficient. If any type of enzyme is present inside a cell for any type of reaction, it will accelerate the rate up to 10 power 3 to 10 power 17 times. Okay. Now, what about the specificity? These enzymes are highly specific. They have high specificity, interacting with one or few specific substance, and catalyze only one type of chemical reaction. Okay, so one enzyme only for one type of substrate, another enzyme for another type of substrate. Okay, so they are having very highly specific interaction with substrate. Third is mild reaction condition. For example, for human being, it is the 37 degree centigrade temperature type, and its physiological pH, whatever it is mild specific. And if we are talking about the range, then between 30 to 40 degrees centigrade, we can say temperature type, and pH nearly slightly acidic or slightly basic, we can say. But yes, like the pH of our buccal cavity is different than our stomach, and that is comparatively different to other parts. So at in each organ different type of pH is there, and on that particular type of, like if acidic pH is there, then only those enzymes that are able to work efficiently at acidic pH, they are only doing their performance there. Okay, some alkaline enzyme are not able to show it their function in acidic pH, right? So but overall their mild reaction conditions, like optimum is 37 in case of human being, but there is a range 30 to 40, and similarly ambient atmospheric pressure and physiological pH. Okay, so enzymes are highly efficient. They are very specific for their substrate type, and they work in a mild reaction condition. But there is one difference between enzymes and inorganic catalysts. Normally, the inorganic catalysts work efficiently at high temperature and high pressure, means above 40 degrees centigrade. Means above 40 degrees centigrade means high temperature means 80, 90, 70, 60, 65, like this. But if optimum temperature range is not there, that is suitable condition for enzyme, that time enzyme get damaged or enzyme get denatured, and they just lose their catalytic activity. Okay, as you know, at high temperature mostly enzymes are proteinaceous in nature, right? At high temperature they get denatured. Okay, so enzyme get damaged at high temperature above 40 degrees centigrade, because within range, already I told you what is the range, optimum range, 30 to 40 degree centigrade for normal enzyme activity. But there are certain organisms which are present in hot water spring, hot vent, sulfur springs, like archaebacteria, and highly cold. Those some archaebacteria also found in Arctic region, where minus 200 degrees centigrade temperature is there, minus 100 degrees centigrade. So those organisms adapt themselves according to their environment. So the enzyme present in their body, in their cell type, are also adapt in that particular situation. So they can catalyze their reaction at that high temperature or extreme, or we can say extreme conditions, means 80 to 90 degrees centigrade. But this is in a unusual situation, or we can say these are certain exceptions. But normally enzyme get damaged at high temperature. Okay, now, so how do enzyme bring about such high rate of chemical conversion? Normally, what happen, as you can see in this particular slide, enzyme actually lower the reactions activation energy. Lower the reactions activation energy. As you can see, this is a graph. On x-axis, progress of reaction is there. This is the x-axis, right? And this is your y-axis. Y-axis, what is there? Potential energy, right? So, if enzymes are not present in a particular type of reaction, that time the any type of substrate which is having a kind of potential energy in it, it need this much amount of energy, to convert itself into the product. Okay, this substrate is having a certain kind of potential energy in it. Okay, so this substrate need this much energy to convert itself into a product. Okay, so this much activation energy is required without enzyme. But if enzymes are present in a reaction system, then those enzymes actually lower the activation energy. This blue line. Okay, so at this time, substrate only need this much energy to come on a transition state and then convert into the product. Okay, so what do enzyme do actually? Enzyme actually lower a reaction's activation energy. All chemical reactions have an energy barrier. You know this very well, right? And this energy barrier is called activation energy, which actually separate the reactant and the product, right? So, activation energy is the amount of energy needed to disrupt the stable molecule type, so that reaction can take place. Okay, so every type of substance, whether that conversion is an exothermic process or endothermic or spontaneous or whatever, they need to reach a transition state, and to reach their that particular transition state, this state, that substrate need a type of energy or activation energy. Okay, now, so the activation energy is the energy required to start a reaction. Okay, and as you know, enzymes are protein. They bind to a molecule or substrate to modify it or lower the energy required to make it react. Okay, make it react. So, what is the role of enzyme? They actually lowering down the activation energy. So very quickly or very easily, substrate get converted into the product. Okay. Now, there are two type of hypothesis. First one is the lock and key hypothesis. This hypothesis was given by Emil Fischer. Another one is the induced fit theory. And as lock and key is known as hypothesis because all the points given in this particular hypothesis is not clear all type of doubts. Okay, while most acceptable one is the induced fit, that's why we can use the term theory for this. Okay, so induced fit theory for substrate binding, this one is given by the Hushland. Okay, so first, lock and key model or lock and key hypothesis. This hypothesis suggests that substrate is completely complementary in the shape to the active site. If this is a type of enzyme, this brown one, this enzyme is having two different active sites. Okay, and this substrate is completely complementary type of shape, exactly complementary shape of this active site. that just like lock and key. Each lock is having a specific key. Each lock is not able, we cannot open the lock with any type of key. There is a specificity or complementary confirmation, both the parts, right? Just like that, each substrate is completely complementary in shape to the active site according to this hypothesis. So it fit perfectly into the enzyme and make enzyme substrate complex. Okay, and there is no change in shape of the active site when substrate bind to it. There is no change. Already that type of shape is present in that type of shape is present in the active site of enzyme according to this hypothesis. And after some time, this enzyme substrate complex get converted into enzyme product complex when old bond has been broken down and new bond will form. And as a new bond has formed, this will convert into the product and this enzyme get separate out from the reaction. Okay, so this is these type of steps has been mentioned in the lock and key hypothesis, but there are some doubts, and all the scientists that are present at that time, they have raised down some questions like, all it means only and only those substances are able to bind that particular enzyme who are having that complementary shape, whether it is actual their substrate or not. Okay, so there were so many queries. So after the so many queries and so many doubts, there is another scientist launched another type of theory that is known as induced fit theory. That is known as induced fit theory. And according to this induced fit theory, binding of substrate actually induce the conformational change in the enzyme. Means already that enzyme doesn't have that type of shape. But as the substrate come towards the enzyme, this substrate induce a conformational change which occur inside a enzyme. As you can see, this is a type of substrate and this is an enzyme. The active site is having some other kind of structure. Okay, some other kind of pockets in it, like you can see. But as substrate come towards the enzyme site, it will create or it will induce a conformational change in the enzyme. Okay, so that's how this enzyme substrate complex has been formed. Okay, so that's why it is written over here that first the substrate bind to the active site of the enzyme, fitting into the active site, and binding of substrate induce the enzyme to alter its shape and fitting more tightly around the substrate. The active site of enzyme, now in close proximity of the substrate, break the chemical bond of the substrate and the new bond or new enzyme product complex has been formed, and this is how this whole process going. Okay, now there are some factors which actually determine the rate of enzymatic reaction, like temperature. So normally reaction rate, as we increase the temperature, like this, as we increase the temperature, okay, normally reaction rate on this particular x-axis and here temperature. So as we increase the temperature, initially reaction rate has been increased up to a optimum temperature around, means optimum around 40 degree. But as optimum it cross the optimum activity range, then again if we start rising the temperature, the enzyme activity rate decrease. Why? Because enzyme get denaturated at this particular site. Initially till 40 degrees centigrade, enzyme rate increase, slowly, increase. But after optimum temperature means 40 degrees centigrade, if we again continuously increasing the temperature, then rate decrease due to enzyme denaturation. Okay. Similarly for pH value determine ionization of group in active site and its surrounding. Okay. So constant pH of body fluid is maintained by buffer system. Each enzyme has its own pH. Like intracellular enzyme pH is around 7. Digestive system pH is around 2. Why? Because in our stomach, HCl has been secreted. So highly acidic environment is there. So only those enzymes are able to work efficiently in that particular environment who can tolerate this pH2, like pepsin. Okay. So similarly, what is the type of activator? They actually accelerate the enzyme reaction. They are certain type of metal ions like calcium, magnesium, zinc, manganese. These are certain activators which enhance the uh, enzyme activity. Okay, some inhibitors are also there. Okay, which actually slow down, or we can say stop the enzymatic reaction, because they are showing competitively similarity between the they have some kind of structural similarity with substrate. Okay, so competitive, similar to substrate, they compete for active site. Okay, so if I'll draw over here, like this is a kind of enzyme, and okay, and this one is the substrate. This one is a kind of substrate, and in the media we are also having some inhibitor, and the concentration of inhibitor is more as compared to substrate. Then what will happen? Substrate compete with the inhibitor, and the concentration of this inhibitor is more in the media in that particular reaction. Then inhibitor come and bind to the active site over here, because they both of them mean substrate. This one is inhibitor, and this sky blue one is substrate. Okay, so inhibitor and substrate both compete to each other for the active site, and whose concentration is more in that particular reaction, that is able to bind with active site. Okay, so that's why it is written over here that inhibitor are competitive, similar to substrate, they compete for active site. Other like heavy metal ion, these are strongly bind to important enzyme group. And similarly, substrate concentration. At high substrate concentration, if substrate concentration is very high, at that time what will happen? Enzyme is saturated, because all the active site now fully blocked with the substrate. There is no free active site. So again, if we continuously enhance the concentration of substrate, then what will happen? After some time, saturation is there, means this type of curve we can obtain. Initially, if we, increase the substrate concentration, reaction going up, but after some time, there is a no increase in rate of reaction, because there is a saturation point occur at this point. Why? Because all the active site present in an enzyme is already blocked with a substrate type. Okay, that's why. And this type of curve is known as saturated curve, which we will discuss it in the next lecture. But right now, as we are discussing about factors who's determining the rate of enzymatic reaction, so little bit I just told you a little bit idea over here. So now, as you can see in this particular graph, this first particular graph is for pH. This one is for pH. Okay, on x-axis pH is there, y-axis rate of reaction. Initially pH 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 2, 3, 3, 4, 5, 6, 7, and so on, 8, 9, 10, like this. Okay, so initially if we increase the pH, initially reaction rate will get increase, but up to a certain limit. After that, if again we start putting increasing the pH, then reaction rate will get slow down. Okay, if this reaction cross its optimum limit. Similarly, for temperature, initially rate of reaction increase with the temperature. If we increase the temperature like 10, 20, 30, 40, 50, 60, 70, and so on. Okay, so initially if we increase the temperature, reaction rate is also increased, but up to a certain limit. After that, again if we start continuously increase the temperature, then suddenly a reaction rate drop it down. Why? Because enzyme get denaturate. As you know, enzymes are protein, and all the proteins are very much sensitive towards the temperature, right? And if we why this one is slow down? Because of denaturation? No, because of deactivation. Okay, deactivation. At this low temperature, enzyme get deactivated. Okay, similarly, these two graphs, like if we increase the substrate concentration on the x-axis, if we increase the substrate concentration, initially reaction rate increase according to the as we increase the substrate type, but after some time, if we again increase the substrate concentration in a particular reaction, as all the active site of enzyme is saturated with the substrate, then there is no enhancement you can see in the reaction or reaction rate in that particular case. Similarly for enzyme, if we continuously increase the enzyme, that time this substrate act as a limiting factor. Here enzyme act as a limiting factor, and in this case substrate act as a limiting factor. So these are certain factors which actually determine the rate of enzymatic. Okay, the term enzyme is given by Kohiner. This, and the first of all, what is the name of that scientist who actually work on this enzyme? That is Louis Pasteur. Okay, he worked on this enzyme, yeast, and fermentation related work. So first discovered by Louis Pasteur, but first this term Kohiner has given this name enzyme, right? So these are certain topic which today I have discussed with you in today's video lecture. Remaining topics we will be discussed in next coming lecture. Okay, till then, I hope all the topics which we have discussed till now has been clear to you. If you are still having any query, any doubt, you can write down to me comment box. We will discuss that later. Till then, take care. Bye bye.